चेंज करू नका, आहे तसंच राहू द्या बरं का. तुमचं फक्त तयारी चालू द्या.
चालेल 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 चालेल. ओके. पिन केलंय आता करून घे 2 मिनिटं. हॅलो हॅलो, आवाज क्लिअर आहे सर?
ठीक आहे ठीक आहे ठीक आहे. तिने मला ऐकू आला म्हणून काय
नाही, मी हे काढून ठेवलं होतं ना.
हा ठीक आहे.
ओके सर, इकडे या ना 2 मिनिटं. तुम्ही मला म्हणे की ते नवीन विंडो मध्ये ओपन करायचं. आता मी माझ्या एंडला जर ही विंडो ओपन ठेवली तर
नाही नाही आहे, दिसतं ते दिसतं ते.
गुड मॉर्निंग सर.
गुड मॉर्निंग. This is Dr. Amol Handore.
Yes sir.
Oh, good. I'm Subedar here.
Yes sir.
गुड मॉर्निंग सर. गुड मॉर्निंग सर.
Good morning.
रामनाथ अंदर.
कौन बोलता है?
रामनाथ, डॉक्टर रामनाथ.
Oh, good morning, Dr. Ramnath. How are you, Dr. Ramnath?
Good morning. How are you, sir?
Oh, good,
I'm
good,
fine.
good, 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 good. Good to hear your voice and good to talk to you.
Yes, sir. Yes, sir.
Okay, so how is the meeting going on?
It is starting now.
Okay.
We'll start in 12 minutes.
Oh, sure. I'm ready. You tell me whenever you are ready. No problem.
Thank you so much.
Hear your voice and good to talk to you.
Yes, sir. Yes, sir.
Okay, so how is the meeting going on?
It is starting now.
Okay.
We'll start in 12 minutes.
Oh, sure. I'm ready. You tell me whenever you are ready. No problem.
Thank you so much.
Hear your voice and good to talk to you.
Yes, sir. Yes, sir.
Okay, so how is the meeting going on?
It is starting now. Shortly.
We'll start in 12 minutes. Oh, sure. I'm ready. You tell me whenever you are ready. No problem.
Thank you so much.
Hear your voice and good to talk to you.
Yes, sir. Yes, sir.
Okay, so how is the meeting going on?
It is starting now. Shortly.
We'll start in 12 minutes. Oh, sure. I'm ready. You tell me whenever you are ready. No problem.
Thank you so much.
Hear your voice and good to talk to you.
Yes, sir. Yes, sir.
Okay, so how is the meeting going on?
It is starting now. Shortly.
We'll start in 12 minutes. Oh, sure. I'm ready. You tell me whenever you are ready. No problem.
Thank you so much.
Hear your voice and good to talk to you.
Yes, sir. Yes, sir.
Okay, so how is the meeting going on?
It is starting now. Shortly.
We'll start in 12 minutes. Oh, sure. I'm ready. You tell me whenever you are ready. No problem.
Thank you so much.
Hear your voice and good to talk to you.
Yes, sir. Yes, sir.
Okay, so how is the meeting going on?
It is starting now. Shortly.
We'll start in 12 minutes. Oh, sure. I'm ready. You tell me whenever you are ready. No problem.
Thank you so much.
Hear your voice and good to talk to you.
Yes, sir. Yes, sir.
Okay, so how is the meeting going on?
It is starting now. Shortly.
We'll start in 12 minutes. Oh, sure. I'm ready. You tell me whenever you are ready. No problem.
Thank you so much.
Hear your voice and good to talk to you.
Yes, sir. Yes, sir.
Okay, so how is the meeting going on?
It is starting now. Shortly.
We'll start in 12 minutes. Oh, sure. I'm ready. You tell me whenever you are ready.
Thank you so much.
Hear your voice and good to talk to you.
Yes, sir. Yes, sir.
Okay, so how is the meeting going on?
It is starting now. Shortly.
We'll start in 12 minutes. Oh, sure. I'm ready. You tell me whenever you are ready.
Thank you so much.
सविता ma'am.
Only 13 participants are there. I think we should wait for 5 minutes more.
Savita ma'am,
Subedar sir, yes sir.
kindly start the recording.
Okay. That's my end. Yes, I'll do that.
Savita ma'am.
Only 13 participants are there. I think we should wait for 5 minutes more.
Savita ma'am,
Subedar sir, yes sir.
kindly start the recording.
Okay. That's my end. Yes, I'll do that.
Savita ma'am.
Only 13 participants are there. I think we should wait for 5 minutes more.
Savita ma'am,
Subedar sir, yes sir.
kindly start the recording.
Okay. That's my end. Yes, I'll do that.
Savita ma'am.
Only 13 participants are there. I think we should wait for 5 minutes more.
Savita ma'am,
Subedar sir, yes sir.
kindly start the recording.
Okay. That's my end. Yes, I'll do that. Good morning, Subedar sir.
Good morning.
Shall we start after 2 minutes?
Anytime.
Okay.
No problem. You tell me.
Thank you.
Savita ma'am, kindly start the recording.
Good morning, Subedar sir.
Good morning.
Shall we start after 2 minutes?
Anytime.
Okay.
No problem. You tell me.
Thank you.
Kindly start the recording.
Good morning, Subedar sir.
Good morning.
Andore sir, shall we start?
Yes, madam, we can start now.
Okay. Good afternoon, everyone. Respected principal of our college, Dr. V.P. Gaikwad sir. Respected HOD of the department, Dr. Vikram Kakurte sir, all the committee members, and all the delegates, as well as I can say, all the participants. Welcome back. Today we have an eminent speaker with us, Dr. Subedar sir. Sir has a very rich biodata, but I'll summarize
madam, we
it
can
in a
start
brief. So Dr. N.K. Subedar sir is recently working as a visiting faculty member at the Department of Biology, ICER, Pune. Dr. V.P. Gaikwad sir. Dr. Subedar sir studied the endocrines of fish and obtained his PhD from Nagpur University in 1973. He took a faculty position at the Department of Pharmaceutical Sciences, Nagpur University, and was engaged in teaching animal physiology and neuropharmacology to B Pharmacy and M.Pharm students. Dr. Subedar sir has published several articles, research articles, and serves as a reviewer for reputed research journals. He supervised the research work of 15 students that leads to PhD degree. His research efforts have been supported by varieties of national funding agencies. Sir has actively participated in organizing BSc-sponsored schools in chronobiology and neurobiology, and in teaching programs which are conducted in various universities, including Ashoka. After his superannuation in 2008 from Nagpur University, Dr. Subedar sir moved to ICER Pune to take up new assignments as a visiting faculty to teach animal physiology and neurobiology, as well as to establish new collaborations. So, such an eminent speaker we have today, and let's listen to him. Over to you, sir. Welcome back, sir. Instead of saying welcome back, I should say welcome, sir.
Sir, speaker, sir, unmute करा, sir, unmute करा.
Okay. Is that okay now?
Yes, sir.
Thank you very much for those kind words, and really very happy to be at this college of yours. I had heard a lot about this college, but never got an opportunity to talk to the faculty and the students. So when this invitation came, I said, well, here is my chance and let me do it. So here I am and addressing all of you, and I feel that well, let's make the most of it. And I'm very happy with the meeting that you have been organizing, and it's very good to know that the Department of Zoology is doing these activities. And is that okay now? Department of Zoology has always been having fun. Always. I was a student of Department of Zoology at Pune University. I will not tell you when, because it was much before all of you were born. So long way back, I was a Department of, and then I did my PhD from when this invitation came. So Department of people towards it, they always sound so sweet to me that whenever anything comes from Department of Zoology anywhere in the world, my first instinct is that yes, I'm going to be there. And I see, so you are just Department of Zoology, and that's good enough for me to be here. Okay.
Thank you so much, sir, for your kind words, as well as for giving us time. And it's our golden chance for us to listen to you.
I will not tell you when, because it was much Professor Gaikwad, so long way back, the head of the department, and then I will all of you. And so, okay, let's get started with our talk. Now, what I'll do is I will let me see if I can share my screen.
Yes, present now. Click on
Anything comes from Department of Zoology anywhere in the world. My first instinct is that yes, I'm going to be there. And I see, so you are just Department of Zoology, and that's good enough for
Just nearer to the hand raise. There is one.
Thank you so much, sir, for your kind words, as well as for giving us time. And it's our golden chance for us to listen to you.
I will not tell you when, because it was then I can share. The head of the department, and then I will all of you. And so, okay, let's get started with our talk. Now, what I will do is I will let me see if if I can share this.
Yeah.
Okay, great, good. So this is I have been studying the brain for quite some time, and I find that this is an amazing organ. I don't have to tell any biology student as to why the brain is here, why the brain is so important. Because right now you are talking to me, and I am talking to you. That's all thanks to your brain and my brain. But actually, right now my brain is communicating with your brain, and your brain is brain is of course a very important organ. And I thought it's a good time to take a view as to what we know about brain today in 2021. How did we arrive at this point, and what are the different landmarks or milestones which we have covered in last about almost 120 years, which has brought us to this to some a little understanding of this amazing organ. So that's what I'm going to take. So what I'm going to take you that and hold you by your finger and take you to the amazing journey. Actually, right now my brain is to almost say last the 20th century. So this is going to be the story of the landmarks of the last 20th century. And let us begin with 1900 or something, and then let us move on step by step, and let us familiarize ourselves and revisit some of those exciting moments about the history of our understanding of the neuroscience, and how we came to this point. It's an amazing journey, and let's begin at this stage. Okay. Now what I'm showing you is something which is very familiar, even as school students know that we are looking at a cell. So I'm looking at a cell, and this is such a familiar figure. I know the plasma membrane, I know the nucleus, the nucleolus, the lysosome. Look, this is a typical cell, okay? And this we have been giving about TB as an entire scientific community. We knew about the synthesis of Robert Hooke when he studied the organ cells. He coined the word cell, and then we knew that there's a liver cell, and there's a pancreatic cell, and there's a muscle cell. Amazing journey, slowly, and then different techniques were used to study the cells, and then slowly by 1700, 1800, we were very concerned that there is a cell, and life is made up of a cell, and the cell is made up of nucleus, and blah 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 blah. This was but now look at this image in front of you. This is the cell. This is the cell. Now you go to the next image and ask you whether does this look like a cell? No, it doesn't look like a cell. It looks like I have some white dots here and there, and those white dots give rise to very long processes, and those processes seem to join to one another, and therefore the concept throughout the 19th century was that brain is not made up of cells. No, there are no cells. Then what do you have? You have a network, okay, and somehow that information goes over the network, or some secretions are there, and that is how. So this was the concept that brain is made up of a certain network of neurons. In almost 1900, 1890, this was the concept that no, there are cells everywhere else in the body. Yes, brain, no, it is not made up of cells. It is made up of some network which is continuous with one. Where one body has a process, it is continuous with another body, and that's what the structure of brain that was envisaged in almost 89. You have a good work. Then came this very interesting scientist. We have heard of him many times, and of course he is Camillo Golgi. We know him very well. Why do we know him? We know him because we know the Golgi apparatus or Golgi complex, or there is a Golgi tendon organ which is there in your joints, or there is Golgi. So this Golgi name keeps on coming. Whether you are a his name keeps on coming. So who was this Golgi? He was a great Italian scientist, and he extended from the earlier century to the 20th century, and he was a histologist, and one of his specialization was to put practice with different stains. Okay, so he would take a tissue and stain it, and he was experimenting with the stains, and he had sections in front of him, sections of the brain of an animal, and he was trying to practice whether how does it, can I use some liver, can I use some silver nitrate as a stain? So that was the experiment he was working on, and he suddenly found that there are certain bodies were stained, and he called them as the silver stain, or he called them as the dark bodies. He actually called them as the dark bodies, and then these are some of the so this is the hippocampus of the rat, and with the help of this technique which we call as Golgi technique, we call it as Golgi technique. So he could stain these large bodies, but he still believed that these bodies communicated with one another, and they form a network, and that brain is made up of network of certain bodies. Okay, we are still not using the word neuron. There's a network of bodies. That's what Golgi thought. After he stained the brain with his specialized silver nitrate staining technique. Good. then what happened? There was, and he was an Italian scientist. Now I'll take you across the small ocean and I'll take you to Spain. And in the Spain, we had another scientist by the name Ramon Cajal. Now Ramon Cajal overlapped with Golgi, and as two scientists happened to have at the same time, they were very bitter towards one another. They were not good friends. No, they were not good body. There was a lot of jealousy and politics between these people, but let's not go into that. But then Ramon Cajal, by using the technique which was discovered by Camillo Golgi, he repeated it, and then he came up with some very amazing images. This is the image of the epithelial cell of the cerebellum, and these are the different granules and different cells. And hold your breath, hold your breath. Ramon Cajal said for the first time that, like other cells in the body, the brain is also made up of cells, and those cells are called as neurons. So it's a very funny situation in which, whereas Golgi came up with the technique, he did not come up with the concept of the neuron. No, he did not. That technique was used by Ramon Cajal, and then Ramon Cajal came up with the and what did Ramon Cajal become with a very basic finding? What is it? That brain is made up of neurons, a cell, just as you have a cell in liver and in pancreas. So this is a cell, and this we call as this very simple observation that what brain is made up of a cell, and the cell is called a neuron. This is called as the neuron doctrine, and this neuron doctrine was formed by Ramon Cajal. Okay, and then onwards, means what? From about 1900 onwards, began a new chapter. So modern neurobiology, as we understand today, its foundations were laid about 1900, and it starts with the Golgi technique discovered by Golgi, perfected by Ramon Cajal, and giving birth to the concept that the brain is made up of neurons. For these discoveries, then in 1906, the two scientists together they shared the Nobel Prize. Camillo Golgi and Ramon Cajal. As I said, they were not best of the friends, and the only time they came face to face with one another was in 1906, when they came on the dais to take their Nobel Prize. Okay, and then again they never saw one another. These two names are permanently in the history of neurobiology. Camillo Golgi and Ramon Cajal. And it's a matter of great pride and honor to realize that their respective countries, they actually released stamps in the honor of these gentlemen. So here is the postal stamp was issued in the name of Camillo Golgi. You can see Golgi Liberation. These are the neurons labeled by Liberation by Golgi, and then again in Spain, in the honor of Ramon Cajal, they issued stamps. And more interesting, not only that, they issued currency. Okay, so you can see here this is a currency note of Spain, and this is some 50 pesos or whatever, I don't know the currency. And on this you get the image of a neurobiologist. So we are very happy that a biologist has been honored by having this picture on the currency of Spain way back in 1990 or something. Now, of course, if you say that neuron is a cell and another neuron is a cell, but then the question was, for processing of information, we do the same. So processing of information came much later. So the question was, do the neurons talk to one another? And if the neurons talk to one another, how do they talk to one another? Was a very important question for which we did not have an answer. So what is the question? Here is a neuron, so I'm sure you can identify. Here is a neuron, here is dendrites, there is a long axon, is another neuron, and this neuron has a dendrite, and axon of the neuron is talking to the dendrite, and this connection we call as synapse today. How does the information flow from one neuron to another neuron was a very relevant question in those days, and answer to that question was given by yet another scientist by the name Otto Loewi. Just read the name Otto Loewi. Okay, and He was an Austrian scientist, and this is considered one of the most colorful, and brilliant, and amazing, and almost or thrilling story in the field of neurobiology. You know what is the story? The story is Otto Lowy had a dream. How does the infantry? He was thinking so much about it. You know that happens to you and me also. When you focus too much on the subject, then in the night we generally have a dream which is somehow related to that subject. Something very similar to happen to Otto Lowy. So he says that one night in the year it was sometime in 1921 he had a dream. And then he had a dream, and it's a dream is a dream. Okay, he had a dream and he went back, and then he got up the next day morning and he started wondering what was the dream I had. And he says that it was the most uncomfortable day in his life when he was trying to, because in that dream he had planned an experiment. The night he had thought of doing something, and on the next day he forgot. But he knew that it was something very important. In his diary he writes that it's one of the most uncomfortable days of my life when I kept on wondering and trying to recall as to what the dream was. On the daytime he did not recall. But then another miracle. Next night when he slept back, again he had the same dream. Got up the next day morning, and when he had the dream, he got up from the bed, he went to the lab. That was about 1 in the night. And that 1 in the night, he took a frog, he isolated the heart of a frog, he isolated the heart of a frog, and he put it in a bath, and then he was looking at the heart which was pumping in the bath, and the isotonic fluid was flowing, and he was looking at the heart, wondering and trying to recall his movements. On the day he did not recall. Then take a heart, next night when he slept back, again we had the same dream. So they also do that if you stimulate the vagus, if you stimulate the vagus, it reduces the rate at which the heart is beating. You know, we know the reason behind that, but those days I did not know. So what was the point? He knew that if you stimulate the vagus and the heart, the rate at which heart is beating goes slow, the heart is impeded. But we still don't know. He didn't know as to what was it that was happening between the nerve and the heart. So then what he did was this amazing experiment in which he had one heart, so he killed one frog. From the frog he took a heart, he put it in his bath, and he kept the vagus nerve intact. So this is the vagus nerve. He separated it, this vagus nerve, and then what he did was he put an electrode. It was already known, and when he stimulated the electrode, he was noting that the rate at which this heart is beating. The record is here. Just this is each pulse here is a cardiac cycle. This is called diastole. Okay. Then what he did was, something nobody had done before. He killed another frog, and from another frog he had another heart. That another heart he put in his bath, and then he started the fluid, the biological fluid, not biological fluid, just the isosmotic fluid which was in this bath. He took it into another bath, and this heart was completely isolated. There was no vagus, and he found that when this heart was normally beating, when he stimulated the vagus, then look at this, the rate at which the heart was beating went down, and after some time it was recovered. In this particular case, just as the first heart, the rate at which it was beating went down, look at the other heart. The other heart also, after some time, it also shows some inhibition back. Very simple and extremely elegant experiment which showed that as a result of the release of vagus, some particular chemical was being discharged into the back, just as it was going to the heart, that it was being discharged on the back, and along with the water or along the isotonic fluid, it went to the other heart and the other back, and when the second heart came in contact with that chemical, you got the inhibition. So this was the first clear proof that when a neuron is excited, the chemical is released. That chemical can talk to the cell, the second cell, and in this particular case, the heart cell. And so for the first time, it was clearly demonstrated in 1921 that there is a thing called a neurotransmitter, and that neurotransmitter was acetylcholine. And along with the water, even before the discovery of Otto Lowy, that that acetylcholine is a neurotransmitter which is functioning at the level of neuromuscular junction, you got the intuition. People already knew that acetylcholine is a molecule. Chemistry people knew that. They got it from plant. They knew that it has acetylcholine. So they knew about the chemistry. People knew about the molecule. They knew about the chemistry. They knew about its biological effects. They simply did not know that acetylcholine serves as a neurotransmitter that is released by a neuron. So that was the first. And as we know, acetylcholine molecule is an amazing molecule. Just look at the immense contribution of this molecule has made to our understanding. So just I want to read here. Acetylcholine was the first neurotransmitter discovered, and you know very well it was done by Otto Lowy. People already know the first receptor, the acetylcholine acts on a receptor, nicotinic receptor. The first receptor to be discovered was of acetylcholine. So the first receptor whose structure was determined was acetylcholine. First receptor whose gene was identified and cloned was again acetylcholine. So acetylcholine is a very special place in the history of biology where so many firsts are associated with the acetylcholine as a neurotransmitter, the receptor of acetylcholine, but the gene of all those. So acetylcholine happens to have a very special place in the history of neuroscience. Great. I'll tell you of another great British scientist, and his name is Henry Dale. And incidentally, he was a very good and a very nice friend of Otto Lowy, about which we have spoken. Otto Lowy is, as we have just now seen, is responsible for acetylcholine. Good. Now, Henry Dale was again acetylcholine. Yeah, please. Yeah, please. Is there a question?
Vadane madam, is there any question?
Please feel free to talk. I tell you another.
Vadane madam. Is there any question? No. Okay.
Shall I continue?
Yes.
Good. As we have just now seen. So let's talk about Henry Dale. Henry Dale, he was working in UK, and then he was working on noradrenaline, and the fact that noradrenaline is a neurotransmitter that came after the discovery of acetylcholine by Otto Lowy, but then it was done by Sir Henry Dale, and then Dale gave us a very simple principle, but we called today. He gave it way back in 2021, 22. That time, Sir gave us a principle that is called as, of course, we call it as a Dale's principle. Now, what is Dale's principle? Dale's principle says that here is a neuron, and that neuron uses acetylcholine as a neurotransmitter, and let's call it a cholinergic neuron. Here is another neuron. That neuron uses noradrenaline as a neurotransmitter, therefore let me call it as an adrenergic neuron. And then they'll say that, well, if there is an adrenergic neuron, it's an adrenergic neuron. If the cholinergic neuron, there's a cholinergic neuron, and a neuron will have only one neurotransmitter, and it cannot have another transmitter, and that was the Dale principle. And it gave a solid foundation, we call it a science, when we could, for the first time, classify neurons on the basis of the neurotransmitters which they contain. However, much later, almost 30 years later, we realized that Dale's principle is wrong. Why wrong? Because the same neuron we know today may contain 1, 2, 3, or 4 neurotransmitters. Okay, so the Dale's principle that a neuron will contain only one neurotransmitter is wrong. Eventually, we realize today we know that a neuron may have 4 or 5 different neurotransmitters. Okay, but anyway, that set the tone and that set the foundation of the way in which we classify the neurons, and even today we use that method of classification. So that is the Dale's principle, and I think that's a very important landmark in our history of neuroscience. And then, as a result of the Dale contribution of these two friends, Otto Lowy and Sir Henry Dale, they both shared the prize in Dale's principle that is 1926. Dale for his discovery of the noradrenergic system and the Dale's principle, and Otto Lowy for his discovery of acetylcholine and the cholinergic neurons. At this moment, I must make a mention of even today we use that method Charles Sherrington. So that is, he was a professor working at Oxford. He was a neurobiologist. He was working on the cat brain, and I will tell you, he has amazing discoveries to it. Created one of the most important discoveries at this grade was, I'll tell you very simple. I am sure you can see my hand moving. Okay, can you see my hand moving?
Yes, sir. Yes, sir.
Great. Now, the principle behind the movement of the hand is when I take the hand in this direction, a set of muscles is contracting. Therefore, I am able to move my hand in this direction, and when a set of muscles is contracting, opposite set of muscles is relaxing. Now, when I do the opposite reaction, the opposite set of muscles has to contract, and the first set of muscles has to relax. So there are antagonistic muscles for every motor action. There are antagonistic set of muscles. When one contracts, the other relaxes. When the other contracts, the first one relaxes, and that's how we are able to see my hands. So we are able to walk, talk, and all those great actions we are able to perform. The principle behind the movement very simple phenomena was scientifically demonstrated by Charles Sherrington, and for this discovery, Charles Sherrington was given the Nobel Prize. But why I am mentioning about Charles Sherrington at this particular time is because Sherrington proposed the term synapse, okay, some communication. So there are concept of what synapse is, they did not have. But the synapse word which we use today was coined by Charles Sherrington, and Charles Sherrington for his discovery, not for the word of synapse, but for how the antagonistic muscles are necessary for to perform or undertake any motor action. That those fundamental studies were taken from Charles Sherrington, for which Sherrington was given the Nobel Prize in 1921. It was led by the Nobel Prize. Now I'm sure you have, most of us have heard of Pavlovian condition. We know of Pavlov, Pavlov, but I'm still going to mention about it because this technique is even used today for understanding the phenomena of how learning and memory happens in our brain. What he would do is he was working on dogs, and he was interested in studying the phenomena of salivation. How does the salivation happen in the mouth of the dog? That was the question he was trying to understand. Now I'm sure he realized that so to study the phenomena, he would start the dog for some time, and then he would take the food to the dog. And as he entered, so there was a piece of meat in his hands, and he was wearing a white coat, and then he would go to the dog and offer the food. And then he realized that sometimes even when he was not having the food in his hands, and the moment dog saw him, the dog started salivating. He started wondering, there's no food in my hand, then why is the dog salivating? So he wanted to study the phenomena further. So what he did was this experiment, which is a very famous experiment. I am sure several of you have already studied this. He was saying that when the dog sees the piece of meat, it salivates. Then what he did was, at the same time, now he did another. If you ring the bell, just ring the bell. Does the dog respond? No. But then he did something amazing. He gave even when he was not having the food to the dog, and rang the bell, and did it every day. And then next time he only rang the bell, and there was no need, that dog started salivating, which means there is a profound change has undergone in the brain of the dog, and the dog has been able to associate that if I hear a bell, which I am not going to eat, I am going to eat my food, but if I hear the bell, that bell means that the bell is I am likely to get the food. So this association between the food and the bell has been registered. Certain memory has been constructed in the brain of the dog. This phenomena we call as conditioning, and even today we have hundreds of experiments, hundreds of laboratories where we use what we call as a Pavlovian conditioning to study learning and memory in the rat mouse, and even Drosophila. Okay, so this is a fundamental, and Pavlov was awarded Nobel Prize in 1904. Incidentally, he was the first Russian to be awarded a Nobel Prize, and in his honor, somewhere in Moscow, they have erected a statue of Pavlov, and his favorite dog, and association with the man and the dog is very well reinforced, and this associate with some very fundamental principles of our brain. Okay. I'll take you a little ahead, and I don't know if you have heard, but I'm sure those who have studied about endocrine system and of the pituitary gland and of the hypothalamus, and those who have studied about the neuroendocrine cells in the hypothalamus, I'm absolutely sure some of you have heard these two names. They are two basic, two fundamental, two original, and the names are Ernst Scharer and Werner Scharer. Now, there's a husband and wife team, and they were working in Munich, in Germany, and the time is about a little before the Second World War. And Ernst Scharer was working on fish, but I'm sure the fish, I have given you the image, and the fish is called as toxinous, and commonly we would call it as a mummy chop. So it is a very small fish, not a big one. So he was working on the brain of this toxinous. He was taking sections to the brain, and he was studying the hypothalamus, and he found that, who found Ernst Scharer? What did he find? He found that there are some of the neurons which are very big. Whereas other neurons are normally big. Is what do I mean by big? Whereas most of the neurons are in the range of 5 micron diameter, 6 micron diameter, 7 to 8 micron diameter, the big ones. The large cells are in the diameter of 15 microns, 18 microns, so very 2 or 3 times larger than ordinary neurons. Okay, so and to draw your attention, I will read this image. This is the image of the brain of the fish Larius batrachus. I was working on it way back 40 years, and this is a slide prepared by me where I can show you the neurons which are very large. Similar neurons were studied by Ernst Scharer, and he came up. For the first time, he came up with the micron diameter, the big ones. He said that this is a neuron, which is a secretory neuron. Okay, and those days, today we are very familiar with the concept, but those days people just refused to accept. No, he said that neuron, either a neuron is a neuron or a gland is a gland. There is no such thing as a neuron serving as a gland. There is no such thing as a neuron having a secretory product of that sort of person. People really, Ernst Scharer had a very difficult time. He was rejected by the scientific community, and nobody wanted to accept this concept by Ernst Scharer. And after almost about 8 or 10 years of his extensive experimentation, then slowly the scientists of the community started accepting the concept of a neuroendocrine cell. Therefore, we can consider Ernst Scharer as a father of neuroendocrinology. Okay? Whereas he was working as a neuron serving as a gland. There is no such thing as a neuron having a secretory product of that sort of person. People really, and she worked extensively on the neuroendocrine neurons in the brain of insects, and she has a series of papers, and our fundamentals of the science of endocrinology in insects, the foundation has been laid by Werner Scharer. And at this moment, I feel very happy to tell you that in 1981, I was attending a conference in Hong Kong, endocrinology conference, and there I had the fortune to listen to a seminar delivered by Werner Scharer. Great. Okay, now I'll take you to, I'm still, those days most of the science was only happening in the Europe, America was still coming up, and I'll take you, now I must introduce, and she's another amazing scientist by the name August Krop. Now let me tell you, as a student of zoology, each and everyone must not only know the name of August Krop, but we must read about his life and about his contribution. And as a teacher of zoology, we must also teach. I would actually request the teachers in zoology to conduct a one full lecture on the great contribution of August Krop. I'll tell you why. One basic discovery for which she was given the Nobel Prize was, very briefly. Okay, what am I talking about? I'm talking about, say, muscle. Okay. In the muscle, there is a blood supply. We know about the blood supply. So there is an artery, and there is an arteriole, and then the meta-arteriole, and then the meta-arteriole will have the branches, and we call them capillaries, and the capillaries will reach in the muscle, and in the capillaries there will be water, it will give away oxygen, it will give away fluid material or the glucose, it will pick up carbon dioxide and other metabolic products, and those capillaries will again get together and they'll form the vein, etc. We know very well, we teach it every time. August Krop demonstrated that in a given tissue, not all the capillaries are open at the same time. He showed that some of the capillaries are open now. Now they'll close, and some other capillaries will open, and then some. So all the capillaries keep on closing and opening because just prior to the capillaries there are meta-arterioles, and they have a smooth muscle, and by contraction of the smooth muscle, the flow of blood to the capillaries can be turned on and turned off. A very fundamental discovery with reference to the blood supply to the tissue was made by August Krog. Okay, and for this, of course, August Krog was awarded Nobel Prize in 1980. But why I want to open now they'll close and some of the capillaries will open, and then learn and read about August Krog is for another reason. He gave what we call as the Krog's principle. Now this Krog's principle is something which every zoologist must know. Krog's principle. What is Krog's principle is if you are interested in discovery or understanding any particular phenomena in biology. I am interested in, say, how the insects breathe. I am interested in finding out how that animal reproduces. I am interested in finding out how that animal changes color. He said for finding out any biological phenomena, if you look around, there is a suitable animal model. He gave what he calls as the Krog means if you want to study the circadian rhythm or day and night cycle, of course a very good model is Drosophila. If you want to study the Parkinson's disease, okay, then rat or mouse is a very good model. There are very so if you get what I just ask you a very simple question: what are the different models? I am interested in finding out how that animal reproduces. Biology we start with C. elegans, then I'll go to Drosophila, then I'll go to Apicia, then I'll go to zebrafish, then I'll go to the rat, mouse, rat, and then you see these are all very interesting models, and these models are used because there is a definite advantage. The attention to this advantage was drawn by August Krog. So if you as a student of zoology or a researcher in zoology, if you want to undertake any experiment, or if you want to understand, there are very first ask yourself a question as to which animal is most suitable to undertake that kind of study. And now I will give you an example where the Krog's principle serves a very ideal purpose, and I will draw your attention to the next amazing phenomena and by where the Krog's principle becomes most relevant. What am I talking about? I'm talking about the neuron. Okay, the neuron. And that neuron we know has dendrites and axons, and the axons are myelinated sheath. And then we also know that the axon carry action potential, and there is just an action potential, and it is action potential is the way by which the neurons conduct message from one end to another end, and when the message goes to the tips of the axon, then there is opening of calcium ion channels. Calcium ions come in. As a result of that, the synaptic vesicles are released. Neurotransmitters which is there in the synaptic vesicles is released into the synaptic space. The neurotransmitters travel to the dendrite of the other neuron, and they carry the chemical messenger. We know all that. And that neuron we know as dendrites and axons. When did this research happen? This research happened means what? When did we? Okay, I'm asking you a basic question. When did we learn the basic fact that the neurons are capable of carrying action potential? Or what is action potential? What is action potential? To that we must go to the period of time during Second World War. The research was actually research almost actually stopped. Biology research almost stopped. Second World War, nobody had time to work on animals. Immediately after that, these scientists, we know all that. Okay. Now before that, okay, let me take you back to 1930. Okay. I'm going to ask you a very simple question. In your library or anyone of you have read the book, you have to answer, and you can answer my question. Have you read the book of what is action potential? The Life of Vertebrates. By J.Z. Young. During Second World War, okay. The Okay, no problem. This was a very standard book when I was doing my lessons in masters in zoology. The Life of Vertebrates by J.Z. Young. He was a brilliant zoologist. The picture is there here. So what he did was in 1930. He around that time he was working on the squid. Now the squid is in front of you. Have you read the book? This is the marine mollusk, and he was working on the different systems of this animal, and he had an amazing observation. The observation was that whereas most of the neurons in the brain and the nerve trunk in these animals are small and normal, there are some neurons which are extremely large. Not many, only two neurons are there. One on the left of the brain, one on the right. They are extremely large neurons, and he called them as the giant neurons. What did he call them as? He called them as the giant neurons. So you can see here is the squid, these are the cephalopod, this is the mollusca cephalopod, these are the tentacles, and on this side there are hundreds of neurons are there, but we ignore them. Now call them as the observation was that whereas most of the students and some of these axons are, listen to me, some of the axons have a diameter of about 1 millimeter. So the axon is so large that you don't need a microscope, you can see it with your naked eye. Now, whose observation was this? This was the observation made by J.Z. Young. Okay, and if you take a transfer section of somewhere of the neural trunk here, you find that where are the rest of the small axons, you can see all around. The giant axon is really big, and the diameter from here to here is about 1 mm. So this is the giant neuron. Who discovered this? J.Z. Young just discovered that this animal has some giant neurons. Now you apply the Croc's principle. What is the Croc's principle? For understanding the biological phenomena, there is a suitable animal model. So suddenly this fact was realized by these two scientists called Hodgkin and Huxley, and they realized that they were interested in finding out that where are the neuron conducts its potential. And for doing that, of course, you want to work on an axon. But if you take an ordinary axon or ordinary neuron, the diameter of the axon is 1 or 2 or 3 microns. So small, you can't do anything with it. So they were interested in going to the research of the J.Z. Young, where he had said that you can. So they went to the squid, they would go to the seafront where the fishermen were catching the sea fish. So from there they would pick up the live squid, and then they would bring them to the lab, put them in sea water, and dissect open the giant neurons, and then they did amazing thing in the giant neurons. So you can see here, this is the giant neuron. This is the picture taken by them in 1945. In the axon, they are introducing a metal electrode. Now they would introduce the electrode only because the diameter of the axon was 1 mm. Diameter metal electrode is very small. They could introduce it because J.Z. Young had discovered them. And then they stimulated the electrode, and they realized that there is a sudden change in the electrical current that's picked up by the electrode, and this is their plot. This is the action potential. So earlier when you in the resting condition, it is about -40. Is the -40-50 is the voltage inside. When you excite it within a period of milliseconds, it goes to almost +40, and again it comes down to the baseline. So this is a recording done by Hodgkin and Huxley, and for this, of course, Hodgkin and Huxley were given the Nobel Prize. But the point I want to make is, here is an excellent example of the Croc's principle. What is the Croc's principle? For studying a phenomena, you have a very good, you have to search for the suitable animal model, and here Hodgkin and Huxley just exactly did that. They got hold of a squid, and squid, because of the giant, they could use it, and then they could come up with the discovery of how the action potential happens, and then of course then they come up with what we call as this is a recording done by the voltage clamp method, and then of course we know a lot about yes please. Here is an excellent example of the Croc's principle. Just somebody has
Vivekanand, sir, any question? No, sir, no question.
please tell everybody that I would be happy to take questions right in the beginning. And so they could use it, and then they could come up with a discovery of how interrupt, raise your hand, feel free and ask me a question. Or you ask question to ma'am, and ma'am will pass on the question to me. But I would rather make this two-way discussion rather than only together talking one way. So okay, yes, please.
Yes, sir, sure, sir.
Yeah, please. Questions are welcome. I'm going to wait for 30 seconds. And if the questions are coming from student, I'll even be double happy.
Dear students, dear participants, if you have any question related to the topic, you can ask. Or you can even post your questions in the chat box.
Tell them that Subedar sir will give you an award if you ask the question. Ask question to ma'am, and ma'am will pass on.
Yes, sure. You will get something best award if you ask question.
Discussion rather than only together talking one way. So okay. And if the question is different, you get 2 hours. I know the problem. Everybody has a question, but they are all shy.
Yes, sir. So we have given them freedom to put their questions in chat box too.
Yeah.
You can even, okay, sir, if any question is there, I'll just let you know. Yes, sir.
Tell them that Subedar sir. So shall I continue, ma'am? If you ask.
Sir, yes, sir, please.
Okay. Now, we are continuing with our journey on about the brain, and I'll take you again way back. This is where are we? We are in Germany. Okay. And the name of the German scientist is Brodmann. Okay, I'm sure several of you have heard the word Brodmann. An amazing, he's a neuroanatomist, and he's working on the brain. Whose brain? Human brain. Yeah, how can you work on the human brain? Of course, you can work on the individual is dead. So he would collect the brain, which was not legal those days. Illegally, he would collect the brain from the dead bodies, and then it was something very, very amazing. What he would do is, as a zoology student, we all know the art of microtomy. Okay, we all know. Okay, so what he would do is, he would cut the brain into tiny, tiny pieces, maybe 1 centimeter by 1 centimeter by 1 centimeter. Okay, then he would label the piece so that he would know as to that piece is coming from which part of the brain. So he would have the brain cut into hundreds of pieces, and then he would label every piece so that he would know that piece is coming. Okay. Then what he would do, he would section take the transfer section of that part of the brain, and then he would study the organization of the cortex. Okay, so what is this thing? The histology of the dead brain, of the dead human being, and he is studying histology. And whenever the cortex is, so he realized that the cortex is uniform but not throughout. So cortex has a particular histological structure, then it changes. Then again it is constant, then again it changes, and depending on that, he numbered the different areas of the brain. Now, what do I mean by that? Now, I will draw your attention to this gray area where he has written 10. This means the histological structure of the cortex of this area of the brain is uniform. But if you go from this area to area 9, and if you compare the histological structure of the cortex from area 9, it is different from the area 10. So if you go to the histological structure of the cortex, so please, the cortex is a mammal made up of 6 layers of neurons. It's gray matter, but this depending on the person reported that the structure of the cortex from one end of to another end of the human cortex is not uniform. It goes on changing, and depending on the change, he had called different areas. So area 11 area, 44 area, 46 area, and this became so standard. So it is a very standard nomenclature. And this is area 70 area 37 area 10. So this is how. So this is a cortex. This is based on what? Nothing to do with function. He did not know what is the function of area 6 or area 3. He did not know. He only knew that histologically each area is very distinct and it's different from the adjoining area which carries a different number. hold on this goes on changing and depending on the, and I will take you to about 1950, where this another element is another amazing. His name is Wilder Penfield. Wilder Penfield. He is a neurosurgeon. Where he worked in for some time in UK, then he worked in USA, and then for some time he worked in Canada. So when he was working in Canada, he was a neurosurgeon. Okay, so he opens the skull, he opens the skull of the patient, and then he does it. Why would the patient go to him? A patient would go to those days. The patient of epilepsy, okay, or the patient who is having seizures. As you know, epilepsy is a pretty dangerous thing to happen. And there were techniques those days. We don't use them anymore. There were techniques those days in which you can remove certain part of the brain, not all. You can remove certain part of the brain, and the observation was that you remove certain part of the brain, okay, the patient gets relief from epilepsy. So, who would go to this neurosurgeon in front of you, Wilder Penfield? A patient who is suffering from epilepsy would go to him and tell him that you are suffering from epilepsy. Please help me, and Penfield would do certain surgery on him. And in many cases, the patient would get a relief. So that was a very, what time I'm talking about? I'm talking about 1951 to 1953. That's the time I'm talking about. Remove some part of the brain, not all. You were some certain part of the brain, and the observation. Now listen to this. All the surgeries which were performed on the brain those days, and even today, are performed in an unanesthetized patient. It is very funny. If you want to work on liver or stomach or anywhere, you have to put the patient in general anesthesia. But if you want to work on the brain, you just apply local anesthesia on the skin, and then you give the cut open, and the patient is so you can talk to the patient. Okay, so he could talk to the patient when you are performing the surgery. Number one. Number two thing that he would do is this number two thing that he would do is he would place an electrode, and even today, randomly in some part of the brain, he would give a slight stimulus, and he would talk to the patient. It is very funny. And while he was doing that, he put his, so here is the human brain, and in this brain, he would just apply local anesthesia electrode somewhere in this part. I'm sure it is primary somesthetic artery. So when he put his electrode, some here you could look at this point on the surface of the cortex. When he placed his electrode here, and when he gave a tiny shock, the patient said that I am having a feeling that I am having a certain sensation of touch, or like ants are walking in my hand. So he put a mark on the map that, and then he gave electrical stimulation all along his blue patch, and then the person realized that look, I am getting sensation. So when he put an electrode on this part of his cortex, the person responded that I am having as if some touch is happening at the level of my lips. When he placed his electrode here, and for the first time, find out discrete areas in the brain which were receiving information from discrete areas from the periphery, and now we call them as the primary sensory area. And this primary sensory area, which was for the first time discovered by Wilder Penfield, happened to be area 1, 2, 3 here, area 1, 2, 3 by Corbin and Brodmann. Corbin and Brodmann did not know the function. The function was discovered by Penfield. Okay. And then he was, so this was the first attempt to map areas in the cortex, and then so he mapped, and then okay, then I'll tell you something more. Then he would place an electrode into this area, which is called as the precentral gyrus, and when he placed an electrode here, and when he gave a small stimulus, you know what happened? The patient's thumb started moving. Here, the thumb started moving. Did he ask the patient to move the thumb? No. He just he's just putting an electrode in one part of the cortex, and then he's giving a shock, and the patient cannot control the movement, and then he changed the position of the electrode, and then he was able to map what we call as the primary motor areas of the human brain. Okay, so our first understanding of the functional discrete areas of the brain comes from the studies of Wilder Painfield when he was actually working on the patients and performing surgery on them to give them relief from the epilepsy from which the patients were suffering. And then still in the same time, 1950, 51, 52, 53, and I'm talking about this guy, his name is Molaison. Henry Gustav Molaison. Now, this is an amazing guy. He's not a doctor. He's not a scientist. No, he's not a scientist. He's not a doctor. And what is he? He's a patient. Wonderful, wonderful. So a very important scientific, there's a very interesting scientific progress in which the patient has as much importance as the doctor. And what is this patient? This patient is H. Gustav Molaison, and in the entire neurobiological world, he is famously known as HM. 51, 50 is known as HM. Everybody knows him as HM. What is the story of HM and why is HM so great? I'll tell you very briefly. When HM was this is an amazing guy. Boy of about 10 or 11 years, he had a very small bicycle accident. And what is he? He's a patient. Till then he was very normal. He had a small accident. So after the accident, he started having fits. This is a very interesting epileptic attacks. In which the patient then he grew up in age, and then he also got married, and then he was an electrician by profession. But his fits kept on getting worse and worse, and it became his life was very difficult because every time he would fall off and was very difficult. What is the story of so why he was at the age of 27, which was in the year 1953. Now, this very interesting year, 1953, and HM is 27-year-old. He goes to another doctor. He goes to another doctor, but for the same operation. What is the operation? He says, do something to me and give me some relief from my epileptic attack. So doctor performs an operation, and when he performs an operation, what he really does is he removes some parts of the on the temporal lobe. So we know that we have a frontal lobe, parietal lobe, and on the side we have the temporal lobe. The inner side of the temporal lobe, right and left, are removed. In his case, so this is the brain. You are seeing the brain from the ventral side, and this is the spinal cord. This is the medulla oblongata, and here is the optic chiasma. I'm sure you will recall. And this on the two there on the so the inner part of the temporal lobe is removed. So this is normal brain. This is removed. In the transverse section, you can see the temporal lobe, the medial part. He removes that part. Okay, and then he stitch up, and then he comes out and he recovers. Who recovers? HM recovers. Okay, and then doctor starts talking to him, and doctor comes. His doctor comes. Listening to his very interesting. Doctor comes, and then he asks many questions, and how are you, and good morning, and good evening, and how do you feel, and everything happens, and then he talks very nicely, very cogently. His understanding is normal. His vocabulary is normal. He gives all the responses. The transfer section is when the doctor goes away. Doctor has spent 3 hours with HM. 3 hours with HM on day 1. And then on second day the doctor comes. And when doctor comes, HM asks him the question, who are you? Means doctor has spent 3 hours with HM on the previous day, and when the doctor goes to HM on the second day, HM has forgotten his understanding is normal. Then another psychologist by the name Brenda Miller, she goes and spends 20 years, 3 hours interviewing, 3 hours interviewing HM. And then on second day, the doctor comes in research papers, comes very fundamental fashion about our learning and memory. She reports that in this part of the medial, in the temporal lobe on the medial area, we have a very important part which we call as hippocampus, and hippocampus is very important for formation of the memory. So what are we learning from the experiments which were surgery on the HM? We know that all the experiences experiences about what you call as a declarative memory. Now, let me tell you what is a declarative memory. If I ask you a question, what is the capital of India, you will say Delhi. You are telling me this is declarative memory. If I ask you what is the capital of England, you will say London. That is declarative memory. But if I ask you, tell me how to ride a bicycle, you cannot answer my question because you don't know how to ride a bicycle. You can show me how to ride a bicycle, but you cannot tell me how you ride a bicycle. That form of memory that helps you to ride a bicycle is called as non-declarative memory. And it was very clear that for the formation of, now listen, he could still look, HM could still, he was an electrician, he could still work as an electrician. So whatever memory he had as a, for doing a job as an electrician, it was still given and he could still function as an electrician. That is declarative memory. But if you teach him today that Delhi is the capital of India and ask him after 5 minutes what is the capital of India, he says I don't know. Means his capabilities to have the declarative memory is completely gone, and so we knew for the first time that hippocampus, which is located in the temporal lobe of the human brain, is the primary organ which is where the memory formation with reference to the declarative memory takes place. So this you can read this slide. However, he showed inability to recall the events in daily life, thus indicating profound loss of short-term declarative memory. This is the capital of India, and has a lot of studies. So I'll tell you, since 1953, particularly the learning and memory people who are interested in studying this phenomena, they have been interested in focusing on the hippocampus. I'm taking this simple example from the animal world. We are all zoological people, we are interested in animals, but just to emphasize the phenomena of hippocampus. What you have in front of you is an image of a crow. It's not a crow, it's a relative of a crow. It's a crow, it belongs to the same family. It's a COVID word. It's Lucifraga columbianus, whatever. You get it in Canada and those areas. Now this fellow feeds on the seeds of these plants here, which you can see in the back. And now what happens is the seeds are available during summer. The seeds are not available during winter when it snows all around. So this fellow, when the seeds are available, it collects the seeds and it hides them under the stones. And when the winter comes and their seeds are not available, he goes to now comes the beauty of the problem. Beauty of the problem. He remembers. Now remember, this guy remembers more than 30,000 stones. 30,000 stones below which he has hidden the seeds which he will use as a food eventually in course of time. Look at his memory. Look at his memory. This bird remembers 30,000 stones. When you see this animal and take out its brain, you find that is the beauty of science. Beauty of science. You find that this fellow's hippocampus is twice the size of hippocampus in other birds. Okay, so there is a clear-cut correlation between the size of the hippocampus and the capability of this animal. So hippocampus is not relevant only to you and me as human beings. Hippocampus is a center for the processing of memory in all the animals, including birds and also including reptiles. So reptiles, we don't have much information, but certainly birds and mammals, hippocampus has been profoundly used as a part of the brain that is known to process declarative memory. Beauty of science. You find that with this fellow's hippocampus. Ma'am, can I ask you a question?
Yes, sir.
You have to tell me as to when I have to stop.
Yes, of course, sir. We have till 30 minutes to go. Yes, sir.
You are stretching too far. Right, then it makes.
Okay, enough, sir. No problem.
Okay. Let me talk about this slide and then a couple of slides and then I'll conclude my talk. Okay?
Yes, sir. Sure, sir.
As a question, we are all that is zoology, and I can ask you a simple question. How do we study the memory in an animal? Okay, I want to so a pharmacologist comes to you and says that this look, I have this strong drug here and my drug is
Yes, of course, sir. We have till 30 minutes
then
to
you
go.
say no, I don't believe you. So you say no, let me test it whether my drug can really
Okay, enough, sir.
really
No
help
problem.
with better memory. Okay, let me how do we test it? Okay, for that we have a very fundamental and a very simple experiment which was given by a scientist called Morris. We call it as a Morris water maze. I'm sure those folks in neuropharmacology must have done this experiment. Morris water maze is you have a big tank here, which is about 5-6 feet in diameter, and you fill it half with water, and in that you put a platform. The platform is made of a glass such that if you put a rat there, the rat can't see it. So what you do here is, so this is the point, this is the room here where there's a window, and there's a wall clock, and there's a monitor, there is a chair, and then you release your animal rat in this, and the rat moves, very moves, and finally it finds the rat doesn't like, it's not an aquatic animal, it doesn't like, and then it finds. Now you put the same animal after 5 minutes, and after 5 minutes, and after 5 minutes, you find that after that the animal straightway goes to the platform. What does it mean? This means that the animal now knows, it has a memory with refers to where the platform is hidden, and it directly goes to the platform and it settles on the platform happily, it doesn't have to swim. There's a wall clock, and there's a monitor, there is a chair, you do an experiment on this rat. That is what you do this electrode and burn away the hippocampus of the animal. Okay, so you have regioned the hippocampus of this animal. Okay, all right. So you first you have this is a normal animal, and then it shows it moves around and it finds the place. Now if you region the animal, it fails to remember and it does the same thing, and then it finds the platform. So has this animal learned? Yes. Has this animal learned? No. So here is the time it has taken to find the platform we call as latency. In the control animal, because it remembers, there's less time, so the latency is less, so the time it has taken to get back to the platform is less. But in the case of the animal where the hippocampus is destroyed, you see it takes a lot of time and it does not remember, its cues are helpless, and it keeps on moving around, and then it finds the platform of the top. And then, alright, I think I'll stop my lecture now. Has this animal and yes, if there are any questions, it would be great to answer them. Yeah, please.
Yes, sir. Thank you so much for such a wonderful lecture. And very nicely you have presented it, even the neurobiology, animal behavior, relationship with the nerve cells and different parts of the brain, sir. I think nobody of us was focused and studied the neurobiology in such a deep manner. So thank you so much, sir. So dear participants, if you have any question, you can ask directly, or you can put your questions in chat box. Sir, by the time we'll take some questions.
Yes.
Sir, usually, traditionally what we do is like feeding gold particles to the kids. So does it has any proportion or relation with the increased memory of the kids or not? Or
Okay, you are asking if there is any correlation between giving gold to the children and improving their memory?
Yes, sir.
I get your question.
Is it your participants? If you have any questions, you can ask directly.
All these methods,
or you can put your questions
this is all
chat box.
science. I mean, there's no scientific
By the
basis
time
for
we'll
that.
take some questions.
So this is our traditional wisdom. This is our traditional wisdom, which may be right, which may be wrong. Okay, but we, as a scientist, as a student of zoology or biology, I will not believe it unless it is experimentally proved. Okay, what do I mean? I'll tell you something. I will say that you give that dose to one group of rat, you give it to another group of rat. I'll answer your question. You give it to one group of rat, your special diet which contains the memory enhancing material, and then you disease both the animals in the Morris water maze, and if the animal which has been given your special preparation which shows better memory, then I will be wrong. But the answer is
Yes, sir. Yes, sir.
we have to test it. Don't you? As a student of science, I will not believe, and you should also not believe in the science which we learn, which we have inherited. Okay, more or less, it's a traditional science. But that's not science. Okay, that's not science. If you want to show it, then you must apply Morris water ways, or there are many other ways by which you can test it. Then only you should say that it works. Till then, it is not a perfectly it will not be accessed in science.
Yes, sir. Yes, sir. Thank you, sir. Sir, some questions are from participants. Let's take them. Sujata Magdu ma'am asked a question. Sir, is there a way to control the brain through AI machines?
In the science which we learned, actually there was a program on BBC which I saw yesterday, in which people have shown that they are putting a certain band around their head, and depending on the person there, I'm just telling you what I saw on BBC. I don't know. The person would focus his thoughts, and because of focusing of his thoughts, there is a robot in front of him, and his hands are tied, and he focuses his thoughts. Because he focuses his thoughts, a certain pattern of electroencephalogram is generated in his brain. That pattern is picked up by the band that is tied around his brain. It picks up that information, and that information is translated and it is taken to the robot, and the robot starts working. I'm just telling you what I saw. Yeah, I don't know. So this is an experiment I have seen on BBC, but I don't know much about it.
Okay, sir. Thank you, sir. One more
Question there, sir. Sir, that means whatever we study, it is a hippocampus which stores it. So, how can we increase our memory of storing information which we read? This is a question from a student.
Okay, very good. Point number one. I did not say that hippocampus is a complete storage of memory. I did not say that. Memory is stored in your entire. Memory is stored in the entire cerebral cortex. For the formation of the memory, information must pass through hippocampus. So hippocampus is like a sort of funnel. If only the information goes to hippocampus, then it can be stored in other parts of the brain. Okay. Having said that, can you repeat the question? Very good. Point number one.
Yes, sir. Yes, sir. The another one, the next point was, so how can we increase our memory of storing information which we read?
Okay. This is the question which every mother of a 12th class standard student will ask. What can I do so that my son or daughter can remember better? Okay. To put that question again, here is okay. The answer to that question is keep on using your brain. Okay. Your brain remains sharp. Okay, that's one thing. And we all know that. If you want to solve a problem, you keep on solving it, then you practice and then you see. But that's the only thing. That's the only way. You practice any particular question, then you are because fact also remains that our brain can rust, so that you know very well what I mean by rust. Okay, so if you don't use your brain, it will rust. But and then there are certain researchers. Again, I will let the name of the scientist University, New York University, and researchers tell us that start doing physical exercise. Listen to this, is very important that I think you all know that to all of us. 20 minutes of rigorous exercise or even yoga is beneficial to sustain our memory. Because fact also remains that
Yes, sir.
our brain can very well what I mean by
Okay, sir. Thank you. Few more questions are there, sir. One is from Anshu. Sir, can we prepare artificial chip to work like hippocampus?
As of today, no. At the present, what we have, all of us, we have chips which can be inserted in certain part of the cerebral cortex of the monkey, and by giving certain stimulation by selected electrodes, we can control the behavior of the monkey.
Okay, sir. Thank you.
That we have gone that far, but implanting a chip in the hippocampus maybe after 20 years it is possible to do it just out of question. It's not out of question. That we may be able to reach there in about 20 years. As of today, no.
Okay, sir. One more question from Pooja Roham. Which area of the brain is affected during stress and depression?
By giving certain stimulation by selected different areas are affected. All right, let's see. That we have Well, every part of brain is susceptible. Okay, no
Yes,
part
sir.
is no. Yeah. For example, we all know. I mean, I can actually question you if you go and you find out as to what are the different causes for which a patient may suffer from Alzheimer's disease.
One more question
Okay,
from Pooja.
the reason is again stress, and the reason is again anxiety. Or you can take another example. You take any dementia, or you take any neurodegenerative disease. Okay, there are as a result of stress for many reasons. One of the reasons is generation of free radicals. They can damage the neurons. Under different conditions, different sets of neurons can be damaged. So it's very difficult for me to say as to which set of neuron will be damaged. But we know very well that in case the dopaminergic neurons of the midbrain are damaged, then the patient is likely to suffer from say Parkinson's disease, or certain basic ganglia are damaged, then the person is likely to suffer. Certain cholinergic neurons are damaged, then the person is likely to suffer from Alzheimer's disease. Those correlations are very well established in the literature. But as a result of stress, and even in the cortex, and we all know that you and I suffer from the area that when the neurons of the cortex they die or degenerate, the first thing that happens to each one of us is that we start forgetting names. Okay, we all suffer from loss of memory damage as we grow in age.
Okay, sir. Okay, sir. Sir, one more question. It is little twisted. Because of the accident, is it possible that someone's memory may enhances or may triggers? Because often we said that because of the accident, memory losses there or part of brain is get damaged.
I have heard of loss of memory. I have not heard of memory when the neurons of the cortex.
Yes, sir.
I think that is little too dramatic to happen. Each one of us
Yes, sir.
start forgetting names.
Okay. Sir, one more question is there. In case of old age people, some of them they are having some kind of a habit like moving of their fingers or moving of their feet if they don't want to do so. Is it possible that so why this happens, sir? Enhances or may triggers?
See, I think what happens I can try. I don't know whether I can answer, but I can try to answer that question. Loss of memory. The fact is that although two things. First of all, number one, neurons don't multiply. Okay.
Yes.
Now this is the statement I am making. Neurons don't multiply. But this statement has two exceptions. There are certain neurons of the hippocampus which multiply, and the neurons of the subventricular they multiply, and these neurons are also contribute to the formation of memory. Okay. Now, having said that, can you repeat your question? I can try.
Sir, my question was like in old age people, they
Okay, so I'll tell you. Okay, it is some number one. Even though we do not get this stock of neurons in most of the other areas of the brain, this statement has two exceptions.
yes, sir.
There are connectivity between one neuron and second neuron and third neuron and then some changes. So the synapses are plastic. You can get my language. The synapses are plastic. Means what? The synapses keep on changing. So one neuron may talk to another neuron by many synapses, but if you don't use that circuit, then the synapses may fall off, they may drop off, they may degenerate, and then one neuron may be separated from another neuron. Or if you do the same exercise again and again, so actually when we say when you are practicing a sport, okay, what you are really doing is you are building certain synapses so that your motor functions happen in a much more regulated and an accurate way. Okay. Now, if a person has developed a particular habit in earlier ages to perform any motor effect, or there is some reason by which some connections are formed, very common in the brain, then involuntarily the person may go on to repeat that action. That is because certain neurons have formed connections with one another, or they may be associated with some other processing in the brain, and if they happen to be activated, then the person will repeatedly undertake that we much more regulated. And
Okay,
I will not answer that question. I will not go into details of that because I also don't know much. But I'll ask you a counter question. There are or I will not give you the answer, but I want all of you to think about it. There are examples when a soldier has gone to the battle and he has lost his right arm. He comes back, he is totally healed, he moves in the society without the right arm, but then he gets very uncomfortable. He goes to the doctor and says that, doctor, my right hand palm is itching. Right hand palm is itching. Does he have a right hand palm?
No, but he has
I will not answer that question.
that sensation somewhere.
Answer. I was getting the question.
Yes, sir.
But he's suffering. Okay, he's suffering. He says my palm is itching. I will not give you the answer, but okay.
Yes, sir.
Has it confused you enough? If it has confused you enough, I can try to give you the answer.
Yes, sir. Because
Okay. You see, although his hand is not there, that area in the brain which used to receive from the hand is still there. Right hand.
Yes, sir.
And now that area is plastic. And that area now communicates with some other part of the brain which is now not communicating with the hand, which works with some other part. And if that other part is somehow excited, the patient confuses that there is itch in my right hand. And those who are interested in this subject, I will recommend a very exciting book called as The Phantoms in the Brain by Ramchandra. Okay, very exciting book. I will strongly recommend reading book if you got that in the brain.
yes, sir. Absolutely satisfied, sir. Thank you. One last question is from Amreen Sheikh. Hippocampus, it just controls our memory, or does it affect our brain functions?
Okay, now your question. Now the question needs a correction.
Yes, sir.
Hippocampus does not control our memory. Okay, no. It processes the neural information so that the memory is formed and stored in other areas of the brain. Okay, does it control the memory? No, it doesn't control the memory. But so that question stands corrected, right?
Yes, sir.
And what is the other part of the question?
Does it affect our brain functions?
Oh, it does profoundly. It does profoundly. Yeah, of course, it does. I mean, you see, it is okay. I'll give you a simple example. If you are okay, you go to the zoo and you are looking at the elephant. Okay, so what is actually? How do you? I'll ask you a very simple question. It's not very simple. How do you know that what you are looking is an elephant? Information so that the memory immediately recognize this is an elephant. How do you recognize the brain? You recognize it because when you, as a child, when you look at the elephant and somebody told you that this is an elephant, you were educated about the elephant. That information was stored. Listen to this. That information about the elephant means what? That it is large. Number one, that it is of this color, that it has plus, that it, all that information is not stored as an image of an elephant. No, your brain does not store the image of an elephant. It stores splits up that information into thousands small bits, into millions, into thousands and thousands of synapses. So there may be a synapse that records the tooth or the tusker of the elephant, or that is the tail of the elephant, and when you really see the elephant, all that information is pooled under the influence of the hippocampus. That information is pooled from those thousands of sources, and then the brain is capable of generating the image of the elephant, and that image matches with the animal in front of you, and then you are able to recognize that, oh, this is an elephant. Does not store the image of the, so there is no control of hippocampus plays a very critical role in storing that information and also in retrieving that information when you want to recall, when you really stand in front of the elephant and ask a question, what is this? And your brain gives an answer, yeah, this is an elephant. And when you really see the elephant,
thank you so much, sir. All of us are truly satisfied with the answers, because these are the things, like all these questions where they might not finding their answers, and you are the expert one in this. So you have given nice answers and even satisfied all of us. Thank you so much for this. Now the Harshada ma'am will going to present a vote of thanks. Over to you, Harshada ma'am.
Good afternoon. Thank you so much, sir, for your great talk and presentation and giving your valuable time for us and our students. I hope, sir, your guidance and the deep knowledge about the neuroscience were much more beneficial and informative for our UG students and teachers to the research students for us. It will be useful for them to find out their opportunities in their careers. And very, sir, team. And I thank you specially impressed from your lecture. And I will request you to take such a session second time. And with permission from our principal, Dr. V.B. Gaikwad sir, and the HOD, Dr. V.R. Kapute, and coordinator, to declare this session is over. Thank you much, everyone, and thank you so much, sir.
Thank you. Very good, sir.
Thank you, sir, for nice talk.
Can I log off now, ma'am?
Yes, sir. Yes, sir, sure.
Thank you.
Thank you specially
Dear participants,